Hi dear students, welcome to one more session of genetics. Today we are going to talk about the chromosomal theory of inheritance. Before going into the chromosomal theory of inheritance, we need to know what we had come across in the previous classes. And of course, you do remember Mendel with his law of segregation, law of independent assortment, and the law of dominance, which he put forward by 1865. He worked on Pisum sativum, and he found that some of the characters of the pea plant were being found to be dominant and some were being found to be recessive. And these factors were being coded, or these phenotypic traits were being coded by certain factors which had two different forms, and he called them as alleles. And among the allele which was being present to be dominant or which was masking the effect of the other was known as a dominant trait, and he put forward the law of dominance. And he noticed that when an individual is containing of two different alleles, and when they form gametes during the process of the formation of next generation, the gametes contained only one allele. That is, the alleles of a particular gene get separated during the process of gamete formation, and that was a law of segregation which he stated. And these segregated alleles uh, in the gametes had the ability to independently assort with the alleles of other genes. They were not restricted that one allele of a particular gene will combine with an only the other allele of this gene. There was no any such restriction. Now, if you go to consider here in the case of YYRR into YYRR, which is the F2 generation of the hybrid which has been formed by Mendel, he noticed that the yellow as well as the round one gamete contained the alleles for capital letter Y and capital letter R, and another gametes were being formed containing capital letter Y and small letter R, small letter y and capital letter R, and small letter y and small letter r. And all these gametes which were being formed, like as we have shown on the x-axis and the y-axis, they have the ability to independently assort with each other and form different types of phenotypes. This was the law of Mendel, or the law of genetics of inheritance, which was being put forward by Mendel in 1865. But the chromosomal theory of inheritance states that it is the chromosomes as well, the ones which have been responsible for the inheritance of genetic characters. And you should remember the journey of from Mendel to chromosomal theory of inheritance is a long one, and it was only in 1900s that around 1902 and 1905 the chromosomal theory was being put forward by two scientists independently, Sutton and Boveri. So let's go through that journey rather than saying what is just chromosomal theory. So if you go to look into the animations provided in the Cold Spring Harbor Laboratory, as I usually take you, you can see that in 1882 Walter Fleming found out the process of mitosis or cell division, and this was Carl Naegeli who produced a technique by which he could stain and view the living cells in 1844. So following the path of Naegeli, the process of mitosis was being discovered in salamander embryos, and these cells were quite big, and then they could see thick chromosomes were being visible by using aniline dyes or staining them by aniline dyes. So when you take a salamander embryo, he noticed that when you put aniline dye, you can stain and you can see the thick chromosomes. And these chromosomes were being quite visible when the cells were being dividing, and the process of mitosis was being discovered, and as the time intervals were being formed, different cells were being formed. So, and we know that it contained various stages such as the interphase, prophase, metaphase, anaphase, and telophase. And during this particular time, you can see that the chromosomes were being arranging at different parts of the cells. In the metaphase, they have been found in the equatorial region, and when the anaphase comes, they have been moving towards the corners of a cell, and finally the cell gets separated out in the telophase to form the daughter cells. So this was the process of mitosis, and we are seeing chromosomes after the work of mitosis was being discovered. So this was mitosis, where a mother cell divides and finally daughter cells are being formed. And the cell theory was also being put forward by Rudolf Virchow long back. And now we know the theory of mitosis, and we watched chromosomes. But even now, the chromosomal theory of inheritance was not been established. We just came to know the process of mitosis, and we visualized the chromosomes during the process of mitosis. Now let's talk about the heroes of the chromosomal theory of inheritance. We have Theodore Boveri. He was quite interested in the chromosomes, and we know what was the method to view the chromosomes. So the development as well as the influence of chromosomes in them was being studied by Theodore Boveri, and in late 1800s he used to work on worms as a model for the study. Now he reasoned that since these worms undergo sexual reproduction, the parent sex cells, the sperm and the egg, must have something which would contribute to the progeny, and this was the chromosomes. Now he mainly took Ascaris, a parasitic roundworm, as his test organism, and he presented a problem that now if you go to consider the sex cells like the sperm and the egg, they should contain, in fact, not the same number of chromosomes as in normal somatic cells, because if they contain the same number of chromosomes as a somatic cells, when a new offspring is being formed, of course, the double number of chromosomes would be formed in the next generation. So he thought that there should be a process that half the chromosome number was being available in the sex cells. And this process is called the meiosis, and they visualized it. And there are two rounds of cell division, as we all know today. It has meiosis 1, which contains of interphase 1, prophase 1, metaphase 1, anaphase 1, and the telophase 1. And we also have another step called the meiosis 2 also. Now, as in the case of mitosis, a cell that is not dividing is said to be in the interphase. And during the interphase 1, the cell duplicates in its nuclear material. And in this example, we are taking a cell which is having 4 chromosomes, which replicate into 8. So, the first stage was the interphase 1. Then after that, you can see now it's getting duplicated. The cell duplicates its nuclear material in the interphase. After that comes the prophase stage, where the chromosome condenses and becomes visible under the light microscope. So it is at the prophase stage that the chromosomes becomes visible under the light microscope. After that, we have the metaphase. Okay, and what is the metaphase stage? So we'll just explore that. Now here, what happens in the metaphase, usually the chromosomes come towards the middle. But before that, in the late prophase itself, what happens? The similar chromosomes, that is a maternal as well as a paternal chromosome, will come and pair. That is, the homologous chromosomes will be formed in the late prophase itself before the metaphase 1 is being started. You know, in the previous slide, we might have noticed that is all the chromosomes are lying at different places. So, in the prophase 1, what happens? The homologous chromosomes will come in pair with each other, or they come together. They form a pair. And in the metaphase 1 stage, these pairs will move towards the cell equator. And that's what we are seeing it. They are moving toward the equator. And then further in the anaphase, what happens? The homologous chromosomes will separate and move to the opposite poles of the cell. This is a what? In the anaphase stage. And then finally, as meiosis 1 ends with telophase 1, and what happens? The homologs will collect at the opposite poles of the mother cell, and the cytoplasm will divide to produce two daughter cells, each with one set of chromosomes. So after the process of nuclear division, you can see the cells are being obtained having the single number of or half the number of chromosomes. Then these daughter cells now will undergo one more round of division, and in the meiosis 2, the centromeres will divide and the sister chromatids separate as they do in the mitosis, and interphase 2, prophase 2 are much shorter in the case of interphase 1 and prophase 1. And further on, you can see that they will separate from each other, and as a result of meiosis, what happens? You get four different types of cells, four different germ cells, which are containing half the number of chromosomes. So, in summary, a cell with two sets of chromosomes at the beginning of the meiosis will divide twice to give four daughter cells, each with one set of chromosomes, and these can further mature into the sperm or the egg. So this was the process of meiosis. Now, he was also working on sea urchins, and he noted that what was the significance of chromosomes was mainly noted when he was working with sea urchins. Now, let's just explore that. What happens is a sea urchin can usually, sometimes it might be fertilized by two sperms. And when this happens, what happens? The first cellular division is uneven, and instead of dividing into the two cells, the fertilized egg will divide into the three cells. So, and further on, what happens? Such sea urchins which were being fertilized by two sperms will eventually die, and they never become into the true sea urchins. So the significance that an egg should be fertilized by the a single sperm was being noted by him in sea urchins. And he noticed that here the set of chromosome is also which is associated with the sperm is also being found to be important here. That is, only one set of chromosomes both from the maternal and the paternal side should be contributed when the process of fertilization has to happen. And simultaneously, in 1902, Walter Sutton, another scientist, and he's an American student at the Columbia University, he also published a paper on the number and the shape of grasshopper chromosomes. And this was his paper showing the different types of chromosomes of the grasshopper, showing that the chromosomes were quite important in the inheritance of genes. In his paper, Sutton lined up the grasshopper chromosomes to show the differences in the size and the shapes, and he realized that the similar chromosomes pair during meiosis, and the segregation of the homologous pairs reduces the chromosome number. And based upon the work, Sutton and Boveri together established chromosomes as a physical basis of the Mendelian laws of heredity, and the segregation of chromosomes during the meiosis was what Mendel had predicted for the segregation of the factors. And so thus two scientists independently found out the chromosomal theory. Sutton as well as Boveri, they worked independently on two different organisms, and they put forward the chromosomal theory of inheritance. So, based upon the work which was being conducted on two different organisms, Sutton and Boveri put forward a hypothesis saying that the chromosomes were the genetic material which were being responsible for the heredity in organisms, and later on this hypothesis was being supported by the work of T.H. Morgan, and it was being accepted as a theory. So what are the features of this? That's what I'm just summarizing. It's this theory, it mainly unifies the theory of genetics, which identify chromosomes as a carrier of genetic material, and it correctly explains the mechanism underlying the laws of Mendelian inheritance by identifying chromosomes with the paired factors required by Mendel laws. And it also states that chromosomes are linear structures with genes located at specific sites called the loci along them. And also we can see that when the cells are being dividing, and these chromosomes also will divide, and they will be passed on to one generation to the next, and this forms the basis of all the genetic inheritance. Of course, you also have to come across the experiments of T.H. Morgan, which gave scientific supporting evidence also for the chromosomal theory. With this, we come to an end. Thank you for now.